but it's better than nothing. And it can help you if you really didn't understand something, you just want to run it back and watch it again. Okay, so endocrine, we start with that. Get rid of this out. Typically in summer, and for the first 2 days, I usually don't have a lab or a quiz the first 2 days because I'm just trying to like get the roster settled. Like you guys are all here today, and maybe someone will be here tomorrow, and people are going to come in and see if they can add. I may add someone tomorrow. Who knows? I don't like to kind of do work I got to enter when the roster hasn't been settled yet. So the first 2 days are primarily presentation, and usually I go for about maybe an hour and a half. Then I take a break. Maybe summer, another hour and a half, but you're looking at a lot of lecture every day in summer. I'm sure you expect that. Maybe you had 3 or 4 hour lectures in 4:30 in the whole semester. You can expect that here too, okay? So, but whatever I do, it'll be uploaded to YouTube, okay? So we start off with the endocrine system. Let me just kind of define the whole system, and then I'll kind of explain this picture. Endocrine system. So, endocrine glands, they synthesize chemical messengers called hormones. Endocrine glands. Synthesize chemical messengers called hormones, which are secreted into the bloodstream. To specific target organs. The hormone elicits some response from the target organ. All right, so I look at that picture and I look at this is kind of the standard introduction paragraph to the chapter. And I would emphasize this word. And you have to know what the gland is. We'll go through identifying them, looking at them under the microscope. I'll go through them, starting with the pituitary gland today. Some gland is secreting some chemical messenger called hormones. So in the picture, it's like the circulating hormone has been secreted into the bloodstream there. And they use the circulatory system as a way so that these chemical messengers can travel long distances to some distant specific target organ. And of course, this is a physiology class. So this is a communication system where the body can maintain homeostasis because the target organs are responding in some way to to maintain homeostasis. Okay? So in your studies, when we go through these different things, what's the gland, what's the hormone, what's the response, okay, what's being regulated there, that's kind of what you have to keep straight in your head. So basically, because they use the bloodstream, hormones are long-distance chemical signals. When you studied cell-to-cell communication in 430, when you studied the synapse, remember studying the neuromuscular junction? Well, those neurotransmitters had to travel a very short distance across the synaptic cleft. But because you can use the bloodstream, you could go from the brain to the kidney, you can go anywhere because blood vessels go everywhere. So they're a very important chemical messenger signal. So basically you got nervous system, you got endocrine system. Those are the two primary ways body cells communicate with each other. So as an example of an endocrine system, well, for example, we have our outline here, gland, hormone, target, and we have this picture here. So, for example, regulate blood glucose. Glucose is the circulation form of sugar in the human body. Glucose is the primary fuel that we use along with fat for metabolism. Well, anyways, let's say you eat a meal of pasta. And that elevates your blood glucose. So in the previous slide, they had a picture of a blood vessel, it's shown as a red tube. This glucose is these little green balls, let's say. And those are circulating in the bloodstream because your digestive system broke it down and absorbed it into the blood. So glucose is circulating, and the levels are elevated. The goal is to get those molecules out of the blood into the cell, so the cell can use it for energy. And it turns out you have, if you look at this figure, they kind of like show the homeostatic level, less than 100 mg per deciliter. But if you eat pasta and it goes up, the imbalance is that the pancreas can detect that elevation. So that's the gland. Now, pancreas has like these cells that respond to elevated levels of glucose. They're called beta cells. I'll put the Greek sign beta here. I'll learn that later. But anyways, in response to elevated glucose, they'll secrete the hormone insulin, if you look at the figure. So insulin, I'll draw insulin as these little blue balls. It'll be secreted into the bloodstream with the glucose. They both are circulating in the bloodstream together. The goal is that insulin will communicate with your body cells that will take glucose out of the blood. Now, they give a few examples there. If you look at the top of the chart there, they got tissue cells and liver. I wish they would list skeletal muscle. So let's list skeletal muscle and liver as the two primary tissues that actually insulin targets. Because remember, you have to have a target for insulin. Insulin is useless unless it has a target to actually take the glucose out of the blood. So liver, I'll liver like that. Skeletal muscle. These are, let's call them insulin-sensitive tissues. They'll present receptors that can actually bind insulin and respond to it. Insulin. So, I mean, to be real simple, okay, let me draw like a cell. Let's pretend it's a liver cell, I guess. And it presents a receptor for insulin. Kind of like on that previous slide, I had a cell like that, right? So that green thing is a receptor for insulin. It's specific for insulin and not other hormones. So if insulin is secreted in appreciable amounts, it will diffuse in a way and bind its receptor. And say the liver cell, it'll immobilize activity that will help take glucose out of the blood. Okay? So I'll use blue because it's going to transport the. I'm sorry. Yeah. Oh, geez. Okay, I messed up my colors, didn't I? Sorry about that. I should use blue because I used green for glucose. So let me fix my boo-boo. Sorry about that. Okay, so insulin, I drew blue, and glucose is green. So the insulin receptor should draw the blue one. Sorry about that. So insulin binds its receptor so that the insulin-sensitive tissue can take glucose out of the blood. So let me just draw glucose. Going into the cell. Glucose is green, insulin is blue. So sorry about the mix up there. But anyways, what's the net effect in terms of blood glucose? You decrease blood glucose. So in this big drawing here, if you go through your outline, put check marks next to it. What was the gland that we discussed that secreted the insulin? Pancreas. The hormone that it secreted was insulin. And the target was the liver and the skeletal muscle. And the response was the decreased blood glucose. Okay, so that's kind of the mechanics of how you should study the endocrine system. And I'll go through everything. Well, for example, I won't go through this as an example, but could you solve the opposite imbalance? Well, you use a different hormone. If blood glucose is too low and you want to raise it, the hormone glucagon would stimulate, and the liver would do the reverse. So the liver is doing this thing where it takes glucose and it stores it to glycogen, and glycogen liberating it to glucose. So let me explain those two words at least, because you may not remember what that is. So I'm going to erase this. Glycogen, that is the storage form of carbohydrate in human physiology. I abbreviate carbohydrate, CHO. Carbohydrate. Which usually is C6H12O6, but I just CHO for short, for carb. And well, basically, if you take glycogen, which is a polymer, it's a complex carb. Think of it as a bunch of monomers chained together. individual hexagon is a simple sugar. But if you link them together, that's a good way to store it in the cells of the liver. Maybe we'll branch out a little bit. So, if you're eating a meal and you want to store the carbohydrate and not burn it, insulin would help you add more individual sugars, call this glucose. And it'll convert it to glycogen just by adding it on. So you're increasing your carbohydrate stores. So you're taking a monomer, glucose, and you're just adding it on the polymer, glycogen, and you're storing it. That's a good way to store it. You got a whole nice store there. Now let's say you exercise and you need to increase the circulation of carb for your workout so you can burn it for energy. So then glycogen, we do the opposite. Sorry, what's wrong with me? It's more glucagon. Glucagon. I call it glucagon your exercise hormone, insulin your rest and digest hormone. The effect of glucagon is to reverse it. You take your glycogen and you convert it to glucose. So you're simply just breaking off a few and you're liberating those so that they can travel in the bloodstream to the working muscles. So those individual carbs will get into the bloodstream. So insulin adds it on, converts glucose to glycogen, and glucagon cleaves them off. It depletes your glycogen stores so that they can be used, they can travel in the bloodstream to the muscle cells that are working. Okay, well anyways, I wanted you to understand that. It's important for this chapter to know the difference between glucose and glycogen is performed by the liver. Well, that was just an example. Are there any questions on that? I want to move on to find some more things about hormones. This whole chapter is about the endocrine system, but the term exocrine also refers to glandular secretions. So I just wanted to contrast those differences. Now for us, the endocrine glands will include all the hormones that we're going to learn, like insulin and glucagon. Exocrine glands, by definition, will not secrete hormones. The hormones are secreted inside the body in the bloodstream. But exocrine glands, they'll secrete things like sweat, tears, right? Exterior to the body, like on the skin surface or something. Or sometimes enzymes. Sometimes they'll be secreted into the GI tract, the digestive tract. So those are examples of exocrine secretions that are glands that don't secrete hormones. And exocrine glands always need a duct to the thing they're secreting into, whether it's the stomach or onto the skin surface. So these have ducts, whereas the endocrine glands are ductless. They don't need a duct. They just need the cells secrete directly into the bloodstream. Okay, so they need a capillary. They're ductless, but they need capillaries. Capillaries are the smallest blood vessels. You know, they're both derived from epithelial tissue. let's remember our 4 basic tissue types: epithelial, connective, muscle, and nervous. Those are the 4 that we taught you in 430. And basically, well, endocrine glands, epithelial tissue cells can secrete hormones, but neurons can secrete hormones too. So I want to write that on here. Neurons. Epithelial cells, there's some neurons in the hypothalamus that also secrete hormones. I want you to know that. Here's a picture of a ductless gland versus a gland with a duct. So they're both derived from epithelial cells in this example. An epithelium lines a surface or a cavity. And for an exocrine gland, if it has an exocrine gland function, the invagination, these glands down here have the secretory function. The glands, the cells of the gland, these are just have a duct function for the exocrine gland. For an endocrine gland, those duct cells have disappeared. And all the cells here, for endocrine function, secrete the hormone directly into the capillary. So what we're going to do is we're going to kind of go through all the glands that are important in human physiology. That's a nice picture of all the glands that we'll talk about. What I want to do tomorrow is to kind of like use the scope that's there and look at all of this, the glands we're going to talk about under the microscope. So maybe this doesn't mean anything to you now, but I'll teach it tomorrow. And your quiz on endocrine histology is on Thursday, so I'm going to get you ready for that. But before I get to the histology, I want to make sure that students fully understand how hormones work. So there's a few things I want to go through, including, I call it factors that affect target cell specificity. So this is kind of the physiology of how hormones work. Some details I want you to know related to that picture. For hormones to work, they have to affect a specific target cell. For hormones to work, they must affect specific target cells. Insulin affected liver and skeletal muscle primarily, okay, but nothing else. And for that to work, consider this. If they're secreted in the bloodstream, I call these the fluid compartments, because blood vessels, vessel means it carries something, it's a container for blood basically. Plasma is the liquid part of blood, and hormones are in there, solubilized somehow, and so that's a compartment. So all the fluid external to the blood vessels and external to the cell is called the extracellular fluid. That's a fluid compartment. All the fluid contained within your cells is a third fluid compartment. There's actually a fourth fluid compartment, it's lymphatics, but I didn't have it on this picture. So what you have to appreciate is, for hormones to work, one factor is the concentration enough? Okay. Is there enough secreted so it could actually diffuse and find its receptor on its target cell? So I give an example, very simple example, enough, not enough. So one factor is hormone concentration. Was enough hormone secreted to get an effect? Obviously, if not enough hormone is secreted, you won't get the effect. And the good news is that hormones are very, very sensitive. And so you only need something like 10 to the 9th molarity, 10 to the negative 12th molarity. That should be enough for an effect. Okay, so you don't need a whole lot to be secreted. I'll just say that concentration is enough. But less than that, well, you may not get a response. Well, the other thing is the receptors. The target cell has to present receptors to the hormone, okay? So usually what happens is, number 2, number of receptors. What you see here is that the target cell will either upregulate or downregulate the receptors to the hormone that it's sensitive to. So, what I want you to know for cells that upregulate the receptor, it does that in response to rising levels of hormone, because it's like the body is trying to respond to it. Now, if the hormone levels stay elevated for too long, you don't want to like overshoot homeostasis. So, the target cell will downregulate in response to persistently high levels of hormone. Okay, so a third factor is the hormone receptor affinity. Hormone receptor affinity. What that means is, well, I give the simple illustration there. If there's a mutation, for example, in the hormone receptor, it won't bind. It has to bind tightly to initiate the molecular biology from a tight fit. So affinity is very important. High affinity is good, basically. Low affinity doesn't work. So those are the 3 factors we usually list the students. So these are hormones, and here's how they'll work well. Okay, concentration, affinity, and if there's enough receptors there. Now, the molecular structure of hormones, most books will categorize maybe 3. I'll just kind of boil it down to 2. Protein hormones and steroid hormones, those are usually the molecular forms we deal with. And it's important to understand that, because proteins basically consist of amino acids, which, well, you know, they're the building blocks of all proteins, including some hormones that we study. So this is the molecular structure of hormones. We won't get into the details of it other than the fact that it's important to know if it's a protein or a steroid. Well, you don't have to memorize the 20 different amino acids. I just wanted you to remember that an amino acid is a carbon surrounded by 4 things: a hydrogen, a carboxylic acid, an amine group, and a functional group there. Okay, so that is the basic structure of an amino acid. And some proteins, some hormones are proteins, and they're water soluble. steroids are lipids. They are not water soluble, they're lipid soluble. That's cholesterol. Most steroid hormones we're going to talk about have that basic structure of 4 tightly interlocked hydrocarbon rings. Okay. So it's the steroid hormones we're talking about. They're classified, steroid is classified a lipid, but they're steroid hormones. And they basically, like I said, are lipid soluble. They're not water soluble. And that's important because they can go right through the cell membrane of the target cell because, well, the cell membrane is made of lipid. So like dissolves like, so they can just go right through and they have this basic structure. That basic structure is a steroid hormone, and I have a couple of examples there. Estrogen and testosterone. There's other ones, but I just wanted to throw a couple of examples of steroid hormones. An example. Estrogen, testosterone. The next unit is repro. I'll talk about estrogen, testosterone a lot more. They have a little crown there because testosterone is the androgen king hormone for men. It's the main one. Now, a third type of hormone that I won't hold you responsible for is eicosanoids. They're derived from lipid, and some books kind of teach that. I have the slide here, but I won't hold you responsible for eicosanoids as a third class. So just write your notes. I'm not responsible for eicosanoids. Please read about them in your book if you like. Eicosanoids. So the reason why we kind of teach the whole protein, steroid, lipid soluble, water soluble, it affects the molecular biology of the target cell. So look at this side-by-side simple illustration. We do want you to know the molecular biology. This is a physiology class. And we keep it simple here. And what we want you to know, the mechanism for, I have steroids first here, I got second there over the board. But anyways, what this is showing you here is, if you're a hormone circulating in the blood and you get to your target cell, but you're a lipid-soluble molecule, you get to the target cell, blue circle target cell, basically. If you're lipid soluble, you go right through. You don't need to present a hormone receptor at the surface because you're lipid soluble. So you could bind your receptor in the cytoplasm. See, there's the receptor for the hormone. Maybe it binds right in there in the cytoplasm. Or maybe you have a nucleus. There's the nuke. And maybe your receptor is inside the nucleus. They call it a nuclear receptor or a cytoplasmic receptor, depending on where it is. And maybe the hormone, the steroid hormone, can bind right directly next to the DNA. Now, I'm not going to try to draw the DNA in there, but the point is, you're binding your receptor inside the cell, okay, because you're lipid soluble. And you're going to turn genes on to make the cell produce some protein to respond to whatever you're trying to respond to. So that's why they call it direct gene activation for steroid hormones. This whole thing here, you're going to turn the genes on. but if you're water soluble and you can't freely pass through the lipid bilayer of the target cell must present the receptor on the surface. And basically, if it's high affinity, it binds, that will trigger the intracellular molecular biology of whatever it is. Okay, so maybe it'll turn genes on, maybe it'll just kind of like do something else. I'll give you a couple of examples. But the point is, the whole field of molecular biology is basically students understanding which molecules communicate with the inside of the cell. For example, G proteins is an example. And that's probably what I'll actually talk about in this class. In both cells, you're trying to get a response from the target cell. You just do it in different ways. You either bind the receptor inside the cell or on the cell surface. So here's a professional picture of it. This is the direct gene activation for a steroid hormone. It's binding its receptor in the cytoplasm. So then that complex can then enter the nucleus and turn on genes. And when you turn a gene on, you try to make a protein to elicit some physiological response. So for that slide. And for the other slide, this is for this one here, where you cannot bind inside the membrane. You have to bind at the cell surface. So they refer to the hormone as the first messenger because you have to communicate with molecules on the inside. So this is worth outlining because it's like there's a lot of molecules here you have to keep track of. So let's kind of outline that for protein hormones. One of the ways they act is through G proteins. So the first step is the hormone binds its receptor on the cell surface for number one. So in this slide, they call the hormone the first messenger. In the second step, on the inside there's a G protein. Receptor activates G sub S. It's a G protein that gets activated and it moves along the inside of the cell membrane. The little G sub S means stimulated. I'll just say G protein activated. Moves along the inside of the cell membrane. Step 3, that G protein will activate a molecule called adenylate cyclase. Activates adenylate cyclase. All right, so what the adenylate cyclase is doing, it's converting ATP to cyclic AMP. Okay. So adenylate cyclase converts ATP to cyclic AMP. just think of ATP. That's your major currency for cell work. Adenosine triphosphate. So I'm going to draw ATP, adenosine triphosphate, 1, 2, 3. So what you can do to get your cyclic AMP, which is the second messenger, you just kind of cleave off one P, cleave off the other P to generate cyclic AMP. Well, the important thing is that the cyclic AMP, that is like the second messenger for this molecular biology. Okay, so you start off with the hormone, that's the first messenger, you get to this, because the cyclic AMP will get the cell to trigger to do all this other work. They listed a bunch of generic terms like activates enzymes, stimulates secretion, opens channels, etc. But that's the cell physiology, that was the goal for the first messenger. Okay. Here's a question I like to ask students to make sure you're not confused. Does the hormone ever enter the cell? That's a yes/no question. What do you think? No. Does the hormone ever enter the cell? No, it doesn't. It acts through that whole cascade of events. There's a good analogy that goes along with this. Imagine you're out for a stroll at night, and you're just walking along, and you see that this house is on fire. So you run up, you pound on the door, and somehow you wake up a small child. And the child comes and says, "Who's there?" And you say, "Oh, it's me, I'm your neighbor, I live down the street, your house is on fire. Open up, let me in, I want to come help." And the child says, "No, stranger danger, I'm not letting you in." She's like, "Okay, fine. Go wake up your parents and call the fire department." And so the child does, and the fire department comes. So anyways, in that analogy, what is guy walking down the street pounding on the door? What does he represent here?
The hormone.
The hormone. Okay. What does the kid represent, you think?
Receptor.
Yeah, it's some kind of, let's say the second messenger, right? It wakes up the parents and does the work, right? So the point is, the hormone never enters the cell. And sometimes I see students kind of fall over on that on exams, and that's the whole point. They have to act through molecular biology. It's the steroid hormone that actually enters the cell. So I want to give you two examples of one protein, one steroid hormone that is important in this class that helps regulate blood pressure, and that's ADH and aldosterone. So what I have here is a picture of kidney physiology. Now I know we haven't had kidney yet, but I think I can walk you through it anyways as an example of hormones working. Sometimes in A&P we do have to refer to things that we haven't gotten to yet. It's too hard to compartmentalize 100%, but I think you can understand this. This is a simple figure. What you see is a kidney cell. So this is the kidney. Okay, inside, well, microscopically inside the kidney, you have all these hairpin microscopic tubules that filter blood plasma and generate urine. But anyways, let me draw a kidney cell in the middle here. Now the kidney cells, they're responsible for generating urine, and basically they help regulate fluid balance. So they're constantly filtering your blood, it's like 70 times a day. They just keep on filtering and filtering. They're trying to filter out toxins in your bloodstream. So when you urinate, you get it all out of your body. And so they're very important for fluid balance and blood pressure. To keep blood pressure up, you need the fluid levels to be right. And so those need to be tightly regulated. So the kidneys can help you do that. And so the kidney cells are responsible for that fluid balance. So on this side, they call it filtrate. You can't see it here, but there's these microscopic capillary filters called glomeruli. They filter all your blood. So what makes it through is filtrate, and you modify the filtrate, it eventually becomes urine. We'll get to the details of that. Just know that this is your filtered blood on this side. And as it flows by kidney cells, and it's flowing that way, down, you have a chance to modify the filtrate so that the body can conserve what it wants, and you don't lose it to the urine. Once you urinate it out of your body, it's excreted and it's gone. Well, the thing here is there's a capillary on this side. It's called the vasa recta. Just know it's a blood capillary. And let's say, let's give a physiological example. You're dehydrated. When you're dehydrated, maybe your blood pressure is dropping a little bit. Now, I know we haven't had cardio yet either, so imagine you cut a blood vessel, a little tube. And you have the vessel wall, and you have the lumen. The lumen is the hole, the space where blood flows. Like if you take a black piece of paper, a thick piece of cardstock paper, and you poke a hole in it, and you hold it up to the light, and the light illuminates through the hole. So that's where they get the term lumen, like light illuminates through holes. Well, anyways, if blood pressure is proper, the blood will exert a pressure on the vessel wall, exerting a positive pressure that you can measure in a patient. And that pressure, if it's high enough in arterial blood vessels, it's enough pressure to push blood to the capillaries of your vital organs like the brain and the heart and the lungs and keep you alive. Now when you're dehydrated and you've lost fluids, doesn't it make sense that blood pressure would drop because you're losing the volume, the blood volume inside the blood vessel, pressure drops. Okay, so that's basically why when you're dehydrated, you might see blood pressure drop, okay, as an example. So if that's the homeostatic imbalance, what can the body do? In this example, they secrete the hormone vasopressin, also known as ADH. It comes from the posterior pituitary. Here's vasopressin's receptor. Here's vasopressin. Also known as ADH. ADH is an acronym that stands for anti-diuretic hormone. That's a main hormone of this chapter you got to know. Maybe you don't know this. A diuretic, say alcohol, does it increase or decrease urine output? Increases. Anything that's considered a diuretic increases your urine output. So what do you think something called anti-diuretic does? Decrease urine output. Isn't that desirable when you're dehydrated? You want to lose less fluids. So what this shows you is, well, first of all, the alternate name, vasopressin. In physiology, whenever you see this in physiology, it means maintain increased blood pressure. Okay, this just always refers to that. All right, so anyways, you're dehydrated. The brain, the pituitary, it secretes ADH into the bloodstream. And it binds its receptor here. Okay, so basically, it's binding at the surface. Therefore, I always ask this on tests and whatnot. Is it a protein or a steroid based on this? Protein. It's a protein. It's going to act through your second messenger. Okay. ADH vasopressin binds receptor. You activate cyclic AMP. That's what it says in this box here. So I'll just put up arrow, increased levels of cyclic AMP. So here's the physiology. Number 3, you insert aquaporin 2 water pores into the apical membrane. Let me write that down. Insert aquaporin 2 into apical membrane. The goal is to take water out of the filtrate and put it back into the bloodstream because you're dehydrated. So normally these kidney tubules are watertight. Now, something took me a while to notice, but you see these like little things right there, these little junctions between cells, little tight junctions. It's like if you took a 6-pack of Coke, you know, like the 6 ring, the plastic rings that hold the Coke cans together at one end. It's like if you turn it on its side. These are like those tight bindings that bind the cells together. It's watertight. So water has to be regulated through the cell. It can't seep in. Okay, it's all regulated by hormones. And so what you got here is these storage vesicles. So that's my little aquaporin, a little water porin. They draw it like this. I don't know why they chose to draw it like that. So that's the storage vesicle with my little aquaporin tube right there. And what they're trying to show you is they stick that little channel right there. So this little divot gets stuck right there. So that's the water pore. And assume we have one on the other side. Boop, boop. So now water can pass through these water pores. Okay, so, and the other thing that you may not notice at first glance, these numbers. 300 milliosmolarity, that's like a saltiness. 600, 700. So let me just write the numbers down. 300, 600, 700. You remember your rules of osmosis? Well, I always teach my students, water goes to where the most stuff is, to where it's more concentrated. Because you're trying to like dilute everything out so it's all equally dilute. So if it's more concentrated here, the rules of osmosis say water wants to go through there. If it's more concentrated here, it's kind of like pulled in to there. It wouldn't happen otherwise because it's watertight. So, or what's the net effect? Let me finish it off here. They write step 4, water is absorbed by osmosis into the blood. Water is absorbed by osmosis into the blood. So the net effect here is, what I like to say to continue on with step 4 is, you increase blood volume, therefore you increase blood pressure, is this. Increase blood volume, increase blood pressure as a result. Not the reverse. How do you increase blood pressure? Because you increased the blood volume by increasing the water reabsorption by osmosis at the kidney. So this is really the physiology, some of the main physiology of today's lecture that you've got to know. Blood pressure is a very common theme, well, in my class anyways, in any 431 class. So the whole idea of blood pressure will come up again when we get to cardiovascular, and it'll come up again when we get to the kidney again. It comes up now in endocrine because we're studying the hormones that do that. So basically, ADH, You know that one. It's a protein. Here's one that's a steroid, aldosterone. Now, aldosterone, I'll get to it, but it's secreted from the adrenal cortex. But it also targets the kidney. It so happens it's right on top of the kidney. And well, here's the cell physiology that I want you to know. So let's kind of go through it. I don't know if I want to draw it out again, but I'll at least go through the steps. You always start with, why do you need the hormone? For this one, it's kind of the same thing. You're kind of dehydrated. Usually when you're dehydrated, I didn't mention this before, of course you've lost fluids, but usually sodium levels have decreased. Low sodium, well, sodium is like Na plus. If you ever wondered why sodium is abbreviated Na, there's a word called natriuresis, which means blood pressure. So this is a molecule that can help increase blood pressure. But anyways, when you have low sodium, like salt, right, and you're dehydrated, I mean, when you lose fluids, it's not, if you can kind of taste your sweat, it's not like water, it's salty, right? So that's kind of why you have low sodium when you're dehydrated. So the goal is, if you're dehydrated, low sodium, low BP, you want to correct all of this. Aldosterone can help. So what aldosterone is doing is, I'll go through the steps. All right, aldosterone binds receptor. You know it's a steroid. So I'll ask you, is it going to bind its receptor inside or outside the cell?
Inside.
Correct, and it's shown right there. It's binding inside the cytoplasm. In cytoplasm. Step number 2. What it says there is this hormone receptor complex initiates transcription in the nucleus. Well, if you remember your molecular your standard cell biology, transcription, translation, get a new protein, right? So step 2 is, I'll just say transcription. You're transcribing the gene, you're reading the code on how to make the protein you need to get more sodium, okay? You transcribe a certain gene that you need. In step 3, in the cytoplasm, you have to translate. Translation. You got to go from, RNA to amino acids, right? You translate the code from nucleic acids to amino acids to get the protein that you want. That's step 3. I said translation. It says new protein channels and pumps are made. So let's kind of like at that new, let's say, sodium channels. You made new ones because you're low on sodium. So in step 4, aldosterone basically, it says aldosterone-induced proteins modify existing proteins. Let's just say you increase activity of sodium channels. You modify it in a way that has the desirable cell physiology for that hormone. Result is increased sodium reabsorption. They say also potassium secretion to keep the charge balance right, but I just want to focus on sodium reabsorption. Step 5. Increase sodium reabsorption. So what the figure shows you is sodium is taken out of the filtrate into the blood. It's very small. I probably can't see in the back. There's sodium there. It's one of the things that's filtered. And if you don't reabsorb it, you lose it to the urine. It goes back into the bloodstream. So it's conserved for the body, and it's not lost to the urine. Okay? Now, I think the physiology term to know is this word here. Reabsorption implies it's absorbed again back into the blood. Where was it absorbed the first time? Through your gut. When you ate the salty French fries and the sodium went into your bloodstream, then your kidney filtered it, and you would lose it to the urine, but then you reabsorbed it. Okay. Now the thing that's not mentioned here that's crucial to understand about this, sodium is a full cation. It has a full charge. For it to be soluble, it is surrounded by water molecules. Okay, it's basically surrounded by water. Now, whenever you pull this molecule, you're pulling water with it. So what most physiology books will say is, when you reabsorb sodium, water follows. And I want you to know that. Because, well, you're getting your sodium replenished because you're dehydrated, but also water follows, so that will in turn increase blood volume, increase blood pressure to help correct the imbalance. Okay. And so those two hormones, they really work together synergistically because they both help correct dehydration. And they both have the same ultimate effect. Except this one also helps reabsorb sodium. Okay. So what we say is aldosterone works very well, and water will follow because of what I just said, but also water will follow if ADH is present, because you'll also get those aquaporins. So I'll put that as a little caveat here. If ADH is present. If ADH is present and you get those aquaporins, you're going to pull way more water. You'll always pull some water because this molecule has to be surrounded by water. Okay, so you'll pull water with it. But if ADH is present, you get the water pores. You'll just pull much more water with the sodium. So those are two examples of the direct gene activation and the second messenger system. You guys been sitting for a couple hours. Let's take a break. When I give breaks, about 20 minutes, but I ask everyone to leave the room. That way I can take a break and kind of leave and go get a cup of coffee somewhere. And so I'll see you, let's say, 10:15.